right, everyone. I think it's time to get started. Good morning, good afternoon, good evening, and for some of you, good night, everyone. And welcome to our webinar series, New Tools of the Trade in Life Sciences, Technology that Enables Breakthroughs. And today's webinar topic is Through the Lens of CyTOF, Resolving Signatures of Stem Cell Aging One Cell at a Time. I'm Renate Weitzbauer, the lead editor of the webinar program here at Bioprotocol, which is an open access online journal with the mission to make research more transparent and reproducible. And before we begin this webinar, let me share a couple of quick announcements. So if you have a question, you can post them in the Q&A section or in the chat section throughout the talk, and Melinda will address them at the end. And the webinar will also be recorded and posted on the website later, so you can revisit it there. Okay. It is now my very distinct pleasure to introduce the moderator of today's webinar, Dr. Javier Rivera Clavé, sorry, almost, who is an instructor in the laboratory of Professor Gary Nolan in the Department of Microbiology and Immunology at Stanford University. And his work focuses on the development and application of highly multiplex technologies to study tissue biology in models of both cancer and infectious diseases, work that has also been published in several renowned scientific journals, including Cancer Cell, Immunity, Nature Communications, and Cell. In addition, he holds 4 patents and received numerous awards, among them the EMBO Postdoctoral Fellowship, the LLS Career Development Award, and just a few months ago, the ERC Starting Grant for proposed work on developing an innovative approach that combines highly multiplexed imaging, in vitro and in vivo tumor models, cell engineering, super resolution microscopy, combinatorial low volume liquid handling, and algorithms for deconstruction of spatial patterns. Javier Rivera Clavé received his PhD in immunology from the University of Barcelona in Spain and trained in cancer biology as a visiting researcher at the Institute for Research in Immunology and Cancer in Montreal, Canada. And with that, thank you very much for being here, and the floor is all yours.
Okay, thank you so much. So today's webinar by Dr. Ermelinda Porpiglia will focus on resolving signatures of cell aging, one cell at a time. So a major question in biology remains how tissues lose function as we age. And a paradigm to address this question has been the progressive decline with aging of skeletal muscle strength and regenerative capacity. Some of these processes are well known, but the mechanisms responsible for age-associated muscle stem cell dysfunction remain elusive. And the major reason for this is because of technical challenges to elucidate functional heterogeneity of the muscle stem cell population at the single cell level. Dr. Ermelinda Porpiglia has developed innovative tools to resolve the functional heterogeneity of the muscle stem cell population. And among those, she has pioneered the development of cytometry by time of flight, a single-cell technology that is capable to analyze 50 parameters in millions of cells. Using this technology, she has been able to resolve functionally and molecularly distinct muscle stem cell subsets that arise with aging and capture stem cell fate decisions in vivo. So I met Dr. Ermelinda Porpiglia when she was a postdoc in the Blau Lab at Stanford, and I can really tell you how meticulously and passionately she did her experiments. So it is no surprise to me that her work has been published in major journals such as Nature Medicine, Stem Cell, Cell Stem Cell, Nature Cell Biology. And well, Dr. Ermelinda Porpiglia is a tenure track now at an assistant professor at Aarhus University in Denmark and an associate fellow at the Aarhus Institute of Advanced Studies. And her research interest is to understand how aging impairs tissue regeneration. I'm very excited to see what will come out of her lab, where she is now continuing her work on resolving signatures of cell aging through the lens of CyTOF. So with this, I would like to warmly welcome Ermelinda, and I hope you all enjoyed this webinar.
Thank you very much, Javi, for the kind introduction. And thank you, René, for inviting me to speak today. It is my pleasure to be here to discuss with all of you how transformative technologies like single-cell mass cytometry really enable us to identify previously unrecognized population of cells, reveal stem cell subsets, and also elucidate novel mechanism underlying stem cell aging. And here you can see in this image of single cell mass cytometry, you can see how the technology can really allow a lens to zoom in into skeletal muscle regeneration and its dysfunction during aging. So today I will talk a little bit about, single cell mass cytometry and how single cell mass cytometry allows us to resolve population of stem cells and progenitor cells in skeletal muscle. And I will talk about the applications of single cell mass cytometry to define identity of myogenic progenitors and resolve signature of muscle stem cell aging and map niche dynamic of muscle regeneration in aging and disease. So my long-term interest has been to really understand the process of muscle regeneration and elucidate a new subset of stem cells that arise during the course of aging. So why am I interested in muscle regeneration? Well, muscle regeneration is something that any of you can relate to. Think about an injury you experience directly or indirectly. We can actually study that in the lab. And we can mimic muscle injury by injecting into the muscle a toxin called notexin, extracted from the Australian tiger snake. So here, then we can follow by histology, we can follow the process of regeneration over time. Here in the injured muscle, you can appreciate how the individual muscle fiber are in a complex and organized structure with nuclei at the periphery. But immediately after an injury, the muscle structure is completely disrupted. 18 hours, there is necrosis of the tissue, and then by day 2, there is already an infiltration of immune cells which come in to clear up the damage and make space for the muscle stem cell to regenerate the tissue. So by day 3, there is the activation of the muscle stem cell, which give rise to progeny, some of which will differentiate and fuse to repair the damage, whereas the remaining will self-renew. And then during the course of the regeneration process, the newly formed fibers will slowly lead to the will allow to completely rebuild the skeletal muscle. So by day 30, as you can see here, there is complete regeneration of skeletal muscle, and the only difference between the uninjured muscle and the injured muscle is the presence of centrally nucleated myofibers. So these really highlight the remarkable regenerative potential of skeletal muscle that can completely rebuild itself after an injury. And the driver of this process are the muscle stem cells. They are also known as satellite cells because of their anatomical location. They reside on top of the myofiber, between the myofiber membrane and the basal lamina. And they are marked by a transcription factor known as FAC7. They were first identified based on their anatomical location in 1961 by Alexander Mauro. And only recently, several groups have been able to isolate the muscle stem cell and perform in-depth studies of their function. So the Blau lab, where I did my postdoc at Stanford University, isolates the muscle stem cell as lineage negative alpha-7 integrin positive and CD34 positive. And they have developed an assay of regenerative capacity where the muscle stem cell can be purified by fluorescence activated cell sorting from GFP luciferase positive donor and then transplanted into the irradiated limb of immunodeficient mice. And their ability to then engraft in the tissue is measured by a non-invasive in vivo bioluminescence imaging that relies on the ability of cells expressing the enzyme luciferase to emit light when they come in contact with their substrate luciferin. And this light can then be detected by a very sensitive camera. So using this approach, the lab was able to show that the stem cell after transplantation, they can expand, self-renew, and then they can also differentiate, giving rise to myofibers that are GFP positive. But the same was not true when primary myoblasts, which is the culture system used to study the late stages of myogenic differentiation, when those were transplanted, there was no engraftment. So the process of regeneration is a quite dynamic process during which the identity of the cells change over time. So as the muscle stem cell become mature myogenic cells and they form new myofibers, they go through a series to the expression of a series of myogenic transcription factors that turn on the myogenic program. As I mentioned earlier, the muscle stem cell express the transcription factor FACT7, and then as they start the differentiation process, as they become activated, they express the transcription factor MIF5, which then is followed by MyoD. MyoD marks the commitment to the myogenic program, and finally, myogenin expression leads to differentiation. So due to different type of studies, we knew that the expression of transcription factor is a cascade that lead to the myogenic program. And this was shown using not only developmental studies, but also lineage tracer and as well as in vitro studies using myoblasts. However, even though we knew that the coordinated expression of myogenic transcription factor led to the myogenic process, and we were able to identify the muscle stem cell based on cell surface marker, the identity of myogenic progenitor cells was not yet revealed or resolved when I started my project. And one of the reasons was the lack of tools to resolve the cell heterogeneity within tissues. In order for the identification of a novel population, it's really required to be able to perform single-cell analysis at multi-parameter. Single-cell analysis enable us to overcome the limitation of bulk analysis, as shown here. So if we were to analyze mixed and complex population and complex tissue by traditional biochemical approaches, we will not be able to pick up the different population that make this complex tissue. However, by performing single-cell analysis, we're able to reveal novel population by protein co-expression pattern. And we can then also identify the signaling dynamics within this different population, as well as molecular phenotype. So I decided to use the myogenic transcription factors as a guide to really identify the myogenic progenitor population based on cell surface marker. However, there was a need for a new technology that would enable us not only to measure multiple cell population, but also to measure multiple parameters within those populations. And when I started my project at Stanford University, there was already a technology that was being developed. And this technology will enable for multiparametric analysis, therefore giving us access to rare cell types, will enable the measurement of multiple molecular phenotypes, phosphorylation, methylation, and acetylation, for example, and will enable the functional analysis of precious samples, just quiescence, proliferation, and differentiation. So this advance in multiparametric single-cell analysis was brought by single-cell mass cytometry, transformative technologies. They use antibodies conjugated to heavy metal to measure up to 50 parameters per single cell. So the cells are stained with metal conjugated antibodies, both on the cell surface and intracellularly. And then they are nebulized to single-cell droplets, which can then be shot through an inductively coupled plasma mass spectrometer, where they're atomized, ionized, and the element composition is then mass measured. And the data can then be analyzed by a variety of different approaches, such as traditional biaxial plot, heat maps, as well as high-dimensional clustering algorithms, such as the X-shift analysis used here. So in order to increase the number of parameters that could be measured, as I mentioned, CyTOF relies on the use of heavy metal isotopes that increase the number of measurable parameters. And it relies really on the use of the lanthanide series of heavy metal. You can see here we have about 35 different isotopes that can be used for conjugation of the antibodies, as well as a variety of different elements that can be used, like the palladium for barcoding, yttrium for antibody conjugation. And then we have other parameters that are used for identifying cellular events, as well as measuring cell viability. And most importantly, in order for us to measure cell cycle, we can use the iodine in 127. So the advantage of single cell mass cytometry compared to flow cytometry are the lack of background, because the single cell mass cytometry really rely on rare earth elements that are not present within the cell, whereas fluorescent flow cytometry has the autofluorescence that is intrinsic to the cells. Then there is the minimal signal overlap, because the elements are very relatively pure, and then they can be detected with high sensitivity by the mass spectrometer. Whereas in fluorescent flow cytometry, there is spectral overlap. And finally, one disadvantage is the destructive nature of single cell mass cytometry, whereas flow cytometry, in particular fluorescence activated cell sorting, allows for sorting of a population. And finally, as I mentioned earlier, the use of iododeoxyuridine for detecting cell proliferation is very useful for performing in vivo studies of cell proliferation, for example. So in order to identify a new cell surface marker that will enable to resolve novel progenitor population in skeletal muscle, I perform a high throughput flow cytometric screening of 176 different surface antibodies. And I perform this screen in both PAC70 screen muscle cells, isolated from a reporter mouse that expressed the screen under the PAC7 promoter, or myoblasts, a culture system used to study the late stages of myogenic differentiation. And then I performed the data analysis looking for markers that were expressed either differentially between the two populations or exclusively in one population and not the other. So an example of a marker that was expressed at different level in the different populations of muscle stem cells or myoblasts was CD9 at tetraspanin, which is expressed at intermediate level in muscle stem cells and high level in myoblasts. Whereas the marker CD104, a beta-4 integrin, was expressed exclusively in the myoblasts. So I decided to use these novel cell surface marker in combination with the myogenic transcription factor to resolve progenitor population in skeletal muscle. And in order for me to do that, I had to develop a whole panel of CyTOF antibodies, because single-cell mass cytometry was applied by the Nolan lab to the study of the hematopoietic system, and therefore most of the reagents available were for the analysis of immune cells and stem cells in the hematopoietic system. So I decided to build my own panel, and in order to resolve the heterogeneity of skeletal muscle, and the key parameters in this panel were the transcription factor, which really enabled me to resolve the whole myogenic compartment. So I'll just walk you through some of the steps in designing your own panel. I know that some of you might be interested in that. When you're developing a panel from scratch, of course, it's important to select the correct antibody clone. And in terms of the cell surface markers, they were already present in the screen, so I didn't have to go through those steps. But for the myogenic transcription factors, I had to detect and select new antibody clones and test them. And then, of course, another important aspect of building a CyTOF panel is to select the right elemental mass tag. And finally, once the antibodies have been conjugated to the metal, then there needs to be a titration in order to select the concentration of antibody that gives you the best signal-to-noise ratio, the optimal signal-to-noise ratio. So when testing different antibody clones, it's important to perform those tests by flow cytometry, usually, and it's important to test them using both positive and negative control. Now, positive control can be a variety of different samples. So primary cells, for example, from reported mice, like the PAC7ZS green, cell lines that express the target protein, or sorted cells. Whereas negative control can normally be primary cells from knockout mice or cell lines that lack the target protein. In terms of selecting the elemental mass tag, so as I mentioned earlier, one of the advantages of CyTOF is the lack of signal overlap or the minimal signal overlap. And this can be ensured by careful panel design. So it is important when selecting a marker, when selecting a tag to assign to a specific antibody, it is important to take into account the expression levels of that marker, as well as the system sensitivity. So markers that are expressed at low level should be detected by antibodies that are conjugated to metals that are in the high sensitivity range, which is between 153 and 175. Whereas markers that are expressed at higher level can be detected by antibodies that are conjugated to metals that are at either end. And so another factor to keep in mind when designing a CyTOF panel is the isotopic impurity. So as I mentioned earlier, there is very little signal overlap, but the isotopic impurity can lead to spillover into the mass +1 channel, whereas the oxide formation that can occur during the run can lead to signal spillover in the M+16 channel. And these, of course, can be needs to be taken into account because the elements that are in the lower end of the spectrum tend to generate oxides. And this is important to make sure that, you know, if the antibodies that are detected, they are detecting highly expressing markers, so it's important that they are taken into account if the marker expression is too high, you know, because there could be spillover in the M+16 channel. So as long as these elements are taken into account, then the panel will lead to minimal spillover. And of course, then as part of the antibody, the panel design is the antibody conjugation step. The conjugation relies on the use of polymer tags that are bound to metal chelators. An example is shown here. So here we have the polymer tag, which is linked to the antibody using a myelamide linker. And this polymer can contain several metal chelator. So the process really relies on the interaction between metal chelating polymer that has been loaded with the lanthanide ions in this case, and the reaction between the metal loaded polymer and the reduced antibody, partially reduced, so that the thiol group can interact with the myelamide linker. And so this will lead to the production of the conjugated antibody. And you know, excess ions can be removed. Can be removed by size exclusion columns. So what's important when conjugating the antibodies is that IgG are usually compatible with the available conjugation protocols that are provided by standard biotools. And the antibody formulation requires antibodies that are BSA and additive free because the BSA can interfere with the conjugation process. So we discussed validation of antibody clones prior to actually conjugation. And here are some data of the validation that I did for my panel, in particular for the transcription factor PAX7. As you can see here, I used for positive control by flow cytometry both soil and muscle stem cells that express the transcription factor, as well as the primary myoblasts, which are not expected in the late stage to express the PAX7. And most importantly, I also used muscle cells isolated from the reported PAX7 green mouse. So as you can see here, we have the PAX7 positive cells, 90% of them are ZS green positive. And then once the antibody specificity has been tested, the antibody can be conjugated, can be titrated, usually with a twofold titration protocol, twofold serial dilution protocol, and then the optimal concentration can be established. And here you can see another additional control that I used was to actually use PAX7 knockout cells to test the antibody. And here in red is the background that comes from the PAX7 knockout, and here is the signal in the wild type tissue. So in addition to metal conjugated antibodies, other reagents can be used to detect cellular events. An example shown here is the DNA metallointercalator used as cellular indicator. So this DNA metallointercalator can be rhodium or iridium based, and they intercalate between the bases of the DNA and can then be used to detect cellular events, because unlike flow cytometry, where we can rely on forward scatter and side scatter to identify cell events and distinguish them from debris, in single cell mass cytometry, we use DNA-based intercalator to actually identify cells when the cloud of ion comes through. And it's important to be able to stain for viability of cells, because when we are focusing our analysis, we want to be able to analyze the live cell population. And for that, cisplatin is actually used. So it's been established by the NONA lab. The cisplatin can be used to measure cell viability. It is a chemotherapeutic, and its chemotherapeutic action really comes from binding to the DNA, whereas the ability to measure cell viability relies on its rapid entrance into the cells that have a compromised cell membrane. And then the cisplatin will bind to protein nucleophiles. And so this staining procedure, the viability staining, is really critical. It needs to be done within a very short period of time, within a minute, and then it needs to be neutralized by serum-containing media. And then finally, I discussed the use of iododeoxyuridine, which it is a nucleotide analog that can be incorporated into the DNA, and it is used for the detection of proliferating cells. As you can see here, it's incorporating into the DNA. It is a great tool to actually be able to follow cell proliferation in the tissue, because it does not require any staining. And as you might know, with flow cytometry, usually the BRDU staining requires denaturation of the DNA, and it can therefore compromise the cell surface staining as well. So we went through the steps to build a CyTOF panel from scratch. That's more or less what I had to do. You've seen some of the controls. And when I finally could build my CyTOF panel, it contained both cell surface marker known to stain or to recognize muscle stem cells, such as the CD29, the CXCR4, CD34, alpha-7 integrin, and VCAM, as well as some of the new cell surface marker identified in the screen. And I also included markers that will enable me to remove immune cells, endothelial cells, and FAP, or fibrodipogenic progenitors. And most importantly, my panel included myogenic transcription factor, PAX7, MIF5, MyoD, and myogenin, which really will enable me to map the myogenic process in vivo. So the muscle was isolated from wild-type mice, digested to single-cell suspension, and then the cells were stained with this metal, with this panel metal conjugated antibody, first on the cell surface, and then intracellularly after permeabilization with methanol. And samples were then run through the mass cytometer, and the data were analyzed using the X-shift clustering algorithm. The X-shift clustering algorithm, it's a k-nearest neighbor. It's based on k-nearest neighbor density estimation in high-dimensional space, and it enables unsupervised clustering of the data sets, therefore allowing the identification of previously unrecognized subsets. I paired up the X-shift clustering algorithm with single self-constructed layout visualization of the data set. And that enabled me to resolve at the single cell level the different clusters. And in this type of visualization, subsets or clusters that are closer to each other in 2D space, they're also similar to each other in high-dimensional space. So this enabled me to identify a variety of different clusters shown here in different colors. When I overlaid on this map the expression of the myogenic transcription factor, I could easily identify PAX7 positive cells. And then I could see that some of the clusters express differential expression of the myogenic transcription factor B5, MyoD, and myogenin. So we know the PAX7 marks the muscle stem cells. Then I overlay the expression of the cell surface marker identified in the screen. And if you remember, the CD9 was expressed at intermediate level in the muscle stem cells and high level in the progenitor cells, so in the myoblasts, whereas the CD104 was expressed exclusively in the myoblasts. And so based on the differential expression of CD9 and CD104, and the combined expression of the myogenic transcription factor, I could infer developmental timing. And I call this different population ST for stem cells and P1 and P2 for progenitor population 1 and 2. Because the myoblasts express high level of CD104 and high level of CD9, we assume that this was the later stage of the progenitors. But of course, we had to validate our finding using functional assays. And in order to do that, I employed the cell transplantation assay. So I sorted the muscle stem progenitor population identified by CyTOF analysis, and I purified them from GFP luciferase donor mice and then transplanted 200 cells into the irradiated limb of immunodeficient mice. And as you can see here by bioluminescence imaging, we could see that the ST population, which I predicted to be the stem cell population, was the one with the highest regenerative potential, whereas progenitor population P1 and P2 exhibited progressively lower ability to engraft in the tissue. So this suggests that the progenitor population P1 was the intermediate between the stem cell and the more differentiated progenitor P2. I then wanted to investigate the response of these different populations to an acute injury. And in order to recapitulate the acute injury model, I injected mice with notexin, which is a toxin isolated from the tiger snake. And then I did that at different time points. And then 8 hours prior to collection of the tissue, I injected mice with IDU. I arrested the tissue and then I performed single cell CyTOF analysis of the skeletal muscle. And here we're looking at a biaxial plot of CD9 by CD104 expression, color in third dimension either by IDU at the top or MyoD at the bottom. So what we could observe based on this analysis was that by day 3 after injury, there was a large expansion of the ST population, followed by a decrease in progenitor population P1 and P2. And by looking at the overlay of IDU and MyoD, we could see that this population was proliferating and also expressing high level of MyoD. commitment marker and activation marker. By day 6, there was an increase in P1 and P2 progenitor cells and an increase also in the level of MyoD. So even though this biaxial type of analysis could enable us to extract some information, we really wanted to be able to gain increased resolution in order to study the dynamics of our population during the course of acute injury. So to do that, I applied the X-chip clustering analysis paired with single-cell force-directed layout, and these enable me to build a molecular map of regeneration. So high-dimensional analysis of acute muscle injury really allow us to resolve the SC, P1, and P2 population based on the coordinate expression of CD9 and CD104 surface markers. So this map was generated using all the time points together. When I dissected this map to the individual time points, I could follow changes in the expression of the myogenic transcription factors. So here, day 0, we could see that the SC population was marked primarily by high level of PAX7 expression and low levels of IDU. And we could see, so what we had observed before, the P1 and P2 population. Interestingly, at day 3 after injury, the SC population, as defined by expression of CD9, intermediate expression of CD9, moved to a totally different location on the map. And this population also showed high incorporation of IDU, whereas the progenitor population P1 and P2 had decreased. And importantly, at day 6 after injury, the SC population, again, as defined by CD9 and CD9 intermediate level and CD104 low expression, was characterized by a subset that was high in PAX7, as well as a subset that was low in PAX7. So interestingly, at day 6, the entire population moved closer to the original location on the map, and the progenitor population P1, P2 had increased. When we looked at the myogenic transcription factor MyoD and myogenin, we could also observe quite a dynamic change in their expression levels. So at day 0, we saw what we had seen in the original map. MyoD was marking primarily the P2 population, whereas myogenin, we could see different levels of expression in the P1 and P2 population. After the injury at day 3, we could see that the stem cell population, as defined by CD9 intermediate expression, showed a subset that was low in MyoD expression and a subset that was high. So there was really a gradient of MyoD expression with the high cells shown here, whereas myogenin expression was still quite low at day 3 after injury. And importantly, this gradient of expression of MyoD was maintained at day 6, with cells expressing high level of MyoD and the population here expressing lower level. But importantly, there was the cells that were expressing high level of MyoD also started expressing high level of myogenin, and this high level of myogenin expression extended from the stem cell population all the way to the progenitors. When we looked at the MyoD low population, the MyoD low population also showed low expression of myogenin. The myogenin expression increased from the stem cell all the way to progenitor P1 and P2, leading to the definition of a myogenic trajectory from stem cell to progenitor cell. So importantly, when we take a look at the entire picture, what we were able to recapitulate was really the process of muscle stem cell activation. So this type of single cell analysis enabled us to resolve the dynamics of skeletal muscle regeneration, where in response to an injury, the muscle stem cell become activated, they incorporate high level of IDU, they start expressing MyoD. So some of the cells express high level of MyoD, whereas others express lower level of MyoD, but importantly, the cells that express high level of MyoD also start expressing high level of myogenin, and this leads to a trajectory that moves from the stem cell all the way to progenitor population P1 and P2. And importantly, the MyoD low stem cells, they start upregulating PAX7, suggesting that these cells are really the cells that self-renew. So in summary, single-cell mass cytometry together with a flow cytometric screen of 176 surface antibodies enable us to identify novel progenitor populations in vivo that are distinguished by differential expression of CD9 and CD104. The novel progenitor population exhibit myogenic potential in vitro and in vivo, and a time course of acute injury by CyTOF analysis really enable us to resolve the state transition, where we could see the MyoD low cells returning to quiescence and expressing a high level of PAX7, whereas the MyoD high cells started expressing the myogenin, and then this myogenin expression extended to the progenitor cells. So we were able to really identify novel progenitor population and elucidate or illuminate the new population. So now I will switch gear and talk about how we apply single-cell mass cytometry to resolve signature of muscle stem cell aging. So as we age, our skeletal muscle mass, strength, and also our regenerative capacity decline. So you can see here in this CT scan of skeletal muscle, you can see the decrease in skeletal muscle mass. And you also, with aging, and you can also see our muscle strength decline over time. This is accompanied by a decline in the ability of skeletal muscle to regenerate as we age. And studies in the Blau lab have shown that if we isolate muscle stem cells from aged mice and transplant them into the irradiated limb of immunodeficient mice, we can see a decrease in their ability to engraft by both bioluminescence imaging and also histology. So these assays, in this assay, 10 cells were transplanted into the irradiated limb of immunodeficient mice. So we're not yet at the single cell level. And this raises the question whether are all the cells losing their function as we age, or are only some of the cells losing their function. So this initial study really raised my curiosity and my interest in really resolving the heterogeneity of the muscle stem cell population during the course of aging. I wanted to understand whether there are specific subsets that arise with aging, and could we identify them? Could we resolve them using cell surface markers so that we could perform in-depth studies of their function? So in order to address this question, I went back to the screen that I had done, and now I focus on the PAX7 screen, muscle cells, the muscle stem cell population. And one of the markers caught my attention was CD47. CD47 was expressed in the entire population of muscle stem cells, but its expression level could distinguish a CD47 low subset and a CD47 high subset. So I included the CD47 marker into my panel, of my CyTOF panel that I had previously developed, and I performed CyTOF analysis of skeletal muscle. I applied high-dimensional X-shift clustering analysis to identify subset of stem cells based on the combination of myogenic transcription factor and cell surface marker, including CD47. And this led to the generation of a map that could identify 3 different clusters. The most prominent cluster shown here in red, the less prominent in blue, and the intermediate abundance in green. I overlaid the expression of the myogenic transcription factor PAX7 and the surface marker CD47 on this map, and as you can see, the most abundant subsets express intermediate to high level of PAX7 and low levels of CD47, whereas the remaining 2 clusters express low level of PAX7 and intermediate to high level of CD47. I then turned to single-cell RNA-seq analysis to investigate the expression of CD47 in young and old and aged muscle stem cells. And here we could distinguish a CD47 low and a CD47 high subset at the RNA level. And then I validate this by flow cytometry. As you can see here, we're looking at CD47 expression in the young in blue and the aged muscle stem cells in red. So CD47 expression increases with aging. And not only the expression level increase, but also the abundance of the CD47 high increase, which is shown here and quantified. So in young skeletal muscle, we have a relative proportion of 60% low, 40% high muscle stem cells. And with aging, the number of CD47 high stem cells increased to 80%. So this raised the question of whether are the functional capability of these stem cells similar or different. In order to address that question, I used the transplantation assay, that's a regenerative capacity, where I sorted CD47 low and CD47 high muscle stem cells from GFP luciferase donor mice that were either young or aged. And then I transplanted 50 cells into the irradiated limb of immunodeficient mice. And in this case, they were transplanted side by side, young and aged, for each population. And then I could see that the CD47 low stem cells shown here, they were actually the one that engrafted best, whereas the CD47 high stem cells showed reduced ability to engraft. And we can see it now quantified here. So independently of whether the cells were isolated from young or old mice, the CD47 low stem cell performed the best, and the CD47 high stem cell exhibited reduced ability to engraft into the tissue. So in summary, by combining the flow cytometric screen, the high throughput flow cytometric screen with single cell mass cytometry analysis, I was able to resolve subset, the rare subset of stem cells that whose relative proportion changed with aging. So the CD47 low subset was the most abundant in young muscle, whereas in aged muscle, the CD47 high subset increased dramatically and exhibited a lower regenerative capability. So this really could explain the decreased regenerative capacity when the entire muscle stem cell population was transplanted from aged mice. Now, I don't have time to go into the details of the functional characterization that I did of the CD47 low and high subsets, but with a combination of in vitro and in vivo assay, we were able to uncover the mechanism, the signaling mechanism by which the CD47 high stem cells impair the regeneration of CD47 low stem cells. They are abundant and they produce a molecule known as thrombospondin, which is a ligand for CD47, and the thrombospondin produced in excess by the high aged CD47 stem cells then act on the CD47 low, which are the functional one, to impair the regeneration process. And by in vivo blockade of thrombospondin using a blocking antibody, we were able to show that we could increase the number of CD47 low stem cells and restore regeneration in the aged mice. So now I'll move on to the current work that is going on in my lab, where we're looking to map niche dynamics of muscle regeneration in aging and disease. So far, what I've talked about has been focused on understanding the myogenic compartment, the heterogeneity of the myogenic compartment, resolving new progenitor populations, and identify stem cell subsets that arise with aging. However, muscle regeneration, it is a multicellular process. If you remember from the very beginning, where I showed the histology time course of acute injury, we could see that immediately after the injury, the immune cells come in to clear the damage. So the muscle stem cells do not act alone, but they really depend on other cells in the microenvironment to be able to exert their function. So immediately after muscle injury, the immune cells orchestrate a transition from an inflammatory phase, which is important for clearance of damaged fiber and dead cells and debris, to a pro-regenerative phase, which is really characterized by anti-inflammatory cytokines. During the end of the inflammatory phase, the muscle stem cells are activated to proliferate, but the pro-regenerative phase is really important for the differentiation aspect of the muscle, the muscle stem cells that have expanded. So we know that there are these two phases, the inflammatory and the pro-regenerative, but we don't know what's driving the transition from one to the other. And we're interested in resolving that, in resolving both the cellular components, the level of immune cells, as well as the signaling aspect of it. And single cell mass cytometry really enables the study of both the cell as well as the signaling dynamic during the course of injury. So the question that we're trying to address is whether the niche dynamics are altered during the course of regeneration in aged tissue. And the approach that we're using is a combination of single cell mass cytometry, suspension cytometry to really quantify the relative proportion of different immune cell types in skeletal muscle, as well as imaging mass cytometry. Because skeletal muscle is a solid tissue, and with single cell mass cytometry, we need to digest the tissue to the single cell suspension. In order to be able to study the process in the tissue, we can benefit from another technology, the imaging mass cytometry, which really enables the analysis of tissue sections using antibody conjugated with heavy metals. And so this approach really enables us to study the relationship between different cell types in skeletal muscle in the native environment, and also investigate how do they interact. So far, with the master student and the PhD student, we have designed a panel that includes a variety of surface markers to resolve the different immune population in skeletal muscle, and we have performed a preliminary study of digested skeletal muscle from young and old mice. As you can see here, we can resolve both myeloid as well as lymphoid immune cells. And we have been working on also resolving the different type of immune cells at the tissue level using imaging mass cytometry. So as you can see here, we have an injury time course of a notoxin injection. At day 0, we can appreciate the individual muscle fiber lined by laminin, shown in yellow, and in red are the CD45 positive immune cells. So you can see there are very few immune cells in an injured muscle, but immediately after an injury, you can appreciate here day 3, there is a lot of degenerating fiber, which are the very small fibers shown here, and they are lined by the laminin. And these fibers are really infiltrated by large number of immune cells. But by day 6, after injury, we can appreciate the regeneration is proceeding effectively. So we have these centrally nucleated myofibers, but there are niches of immune cells that are in a specific location. So what we're interested in is really resolving niches of muscle regeneration, and we're interested in really identifying the different type of immune cells that drive the regeneration process. So we're now working on applying the panel we have developed for CyTOF to imaging mass cytometry in order to address those questions. So finally, we have discussed a lot of the application that I am employing in my work, but I wanted to broaden this up and discuss other applications that can be used for, that the CyTOF can be used for. At the very beginning, I talked about the ability of multiparametric analysis to really study very precious samples. An example could be genetic mouse model or human sample that are very precious. We can perform one experiment, multiparameter, which really enable us to use small amount of material. Studies can also be done in cell lines and 3D cultures. But importantly, single cell mass cytometry started more at the phenotypic level to be able to really identify a variety of different immune cell types and study their signaling dynamics. Now, a variety of other applications are being developed. So we are really focused on resolving function and investigating parameters like metabolism, cell cycle, proliferation, survival, or cell death. You can find a lot of these reagents available, but you can also conjugate your own antibody if you have a specific functional marker that you would like to investigate. And with that, I would like to thank the collaborators, Stanford University. This work was performed in collaboration with the Nolan Lab, in Helen Blau's lab, and some of the collaborators have moved on to different positions now. And finally, I would like to acknowledge my lab that I've recently started at Aarhus University, as well as funding sources from Aarhus University and from the time at Stanford. Thank you all for your attention. I'll be happy to take any questions.
Thank you, Ermelinda. This was a fantastic presentation, and certainly a great example on how the application of single cell CyTOF and even a little bit of single cell RNA-seq, right, can help us better understand the mechanisms of age-associated but also acute injury in muscle stem cell dysfunction. So, well, and I also have to say that I really look forward to see what comes out of the imaging experiments. So I'll start with some of the questions from the chat, but also from the website. So when developing an antibody panel for a new tissue of interest, what is the minimal number of different antibodies to get good resolution? What is your experience with this?
The minimum number of antibodies?
The minimum number of antibodies, I guess, to define your clusters in your population.
So, well, it really depends on the tissue. The ability of measuring up to 50 parameters by CyTOF really enable us to drill down to the population of interest and exclude all the other populations that have been defined phenotypically. So, for building a panel, I mean, the first panel that I showed in the skeletal muscle contained about 23 antibodies, but in terms of like the number of antibodies that then you use for your clustering analysis is much lower, right? Mm-hmm. So I would say in order to get, it really depends on the cell population of interest. If it is a rare population, then you will probably need a lot of different antibodies just to drill down to the population that you want to resolve. And there is really no minimum number, I would say.
Perfect. So what could be the challenge with homemade isotope conjugation, and how can it be improved?
So with the in-house conjugated antibodies
versus commercially buying an already conjugated antibody.
Yeah. Okay, so during the conjugation process, of course, you don't recover 100% of your antibodies. So the recovery usually between 65 and 85%. So you start from a certain amount, and then you end up with less. I guess that would be one challenge is the recovery and the amount of antibody that you get, and then you will have to titrate your antibody to establish the right concentration to use, but that's something that you also will do with commercially available antibodies. Mm-hmm. The other thing is commercially available antibody have already been tested. And they are sure to work. Whereas when performing your own conjugation, as I mentioned, you have to do a lot of pre-conjugation testing by flow cytometry to determine the specificity of your antibody and so on and so forth.
Mm-hmm. So is the antibody used in CyTOF the same that is used in Western blot?
So usually... usually not, because of course it depends. But I think if an antibody has been tested and works in flow cytometry, most likely it will work for CyTOF. And the reason is that with the Western blot, you are denaturing your protein, right? So whereas by flow cytometry and mass cytometry, you are normally testing the protein in their native environment. So you will have, if an antibody has not been tested for flow cytometry, but has been tested for Western blot, or works well for Western blot, you can definitely test to see whether it will work by flow cytometry. But usually antibodies that are used for IP are better suited for flow cytometry and mass cytometry, because can detect the protein in their native configuration.
Perfect. So how long does it take to create a validated panel of antibodies for CyTOF? Is it, a month, a year? What's your experience here?
I mean, it depends on how fast you will proceed, and there are a lot of limitation about the ordering and finding the right antibody clone. But I would say between 3 and 6 months, perhaps, depending on the number of antibodies that you need to test and whether everything is available, right? All your positive and negative controls are there, or do you need to order mice for knockouts? And, I think the time limitation really relies on the availability of the reagents and then how quick you can process your samples.
Yeah.
The test.
So once you have the validated panel, how long does it take to actually perform? When you have your cells and your panel, how long does it take to run an entire experiment, including running the instrument?
Well, that really, once the panel has been validated and the antibodies have been conjugated and titrated, I think something that I didn't discuss during the presentation was that the titration, ideally, you want to do it in the same tissue or the same samples that you're going to finally run at the end, because then you know that you will select the right concentration. But of course, if that's not available, then you can always use cell lines. So going back to the question, it should be very rapid. Of course, it depends on the experimental setup. For me, I need to do the injury time course, and I usually take all week. And then the samples are analyzed, and depending on the number of samples that you are analyzing, you can do that. You can run your stat in the same day or across multiple days.
And is there any challenge on running muscle cells in the CyTOF? Because, those are probably larger cells than typical immune cells that are really, they're smaller and they run perfectly through the capillary. But is there anything that, that is challenging there? And do you have any tips on how to solve that?
Yeah, so interestingly, the muscle stem cells are quite small. But of course, the skeletal muscle tissue, once you digest, it contains a series of different cell types and also, fiber fragments that are some of the challenges. So it is really important to have a very high quality cell suspension when performing the CyTOF. I didn't go too much into the details of the digestion of skeletal muscle, but usually it's a combination of mechanical digestion and enzymatic digestion. So we will cut the tissue either using something like gentle max or scissors, and then combine that with the sequential collagenase in this phase digestion. So having high quality single cell suspension is key. So being able to filter out all the debris, it's one of the tips. So using several filtering steps, and especially prior to fixation of the tissue, of the single cell suspension, I think that would be one of the advice, because otherwise there would be a chance of getting the instrument perhaps clogged. And depending on the time point of injury as well, day 3 after injury, there are usually a lot of there is a lot of cell death, so it's important to make sure that the quality of the suspension is high.
Correct. So Michela Bruschi is asking whether, well, can you label fixed cells or only fresh ones? What's the...
Yeah. So when performing a functional analysis, right, so which require analysis of signaling events, for example, it is really important to fix the cell suspension right away. And it is possible to perform the analysis on the cells that are fixed with the paraformaldehyde.
So what do you typically do in your experiments?
Yeah, so normally, because I've been combining it sometimes with signaling analysis, I fix the single cell suspension immediately after it's generated with paraformaldehyde, and then I proceed to the surface staining. Then I permeabilize cells with methanol and proceed to the intracellular staining. And then, of course, the last step is to fix cells in the DNA, iridium DNA intercalator. And
I guess this
overnight.
Yeah.
Mm-hmm.
I guess this relates to what you were saying before of, titrate your panel in the actual samples that you will run. Because if you
Yes.
validate something, let's say in something that has not been fixed, and then you fix your sample, then you might run into issues. Okay, so you've identified a series of subpopulation in your muscles population. And do you think they can be further subcluster? And do you think that single-cell RNA-seq can help there? Is this complementary, or what are your thoughts on this?
Yeah, so the first part of the talk was more focused on resolving progenitor population in skeletal muscle. And I think that based on the functional assay, the transplantation assays, we have two distinct progenitor populations that are at different stages of the differentiation. Of course, we could try to resolve them further using single-cell RNA-seq. The single-cell RNA-seq really enable us to investigate a really large number of genes. So we could do additional subsetting there. It really depends on what the research question is that the researcher is trying to address. And in terms of the muscle stem cells, of course, yes, we can now isolate the CD47 low and CD47 high muscle stem cell subset, and we know that they're I've combined the single cell mass cytometry with single cell RNA-seq, also to look at, for example, the thrombospondin expression. So we can further characterize, we can further perform further analysis by single cell RNA-seq to gain more functional information about this subset. And it is definitely possible to get more functional information, because with RNA-seq, of course, you're looking at RNA, so you could look at downstream events, downstream of signaling pathways, for example, to see what are the signaling networks that are activated in those cells, and are they different in the young versus the aged muscle stem cells? So there is always more we can do.
Yeah. So what are the most challenging steps of the muscle stem cell isolation process?
I think I discussed a little bit before, right? So we start with the mechanical dissociation, and then the enzymatic digestion. So some of the tips or something that I could point out is, of course, if we are over digesting the tissue, then we risk damaging the cells. So if we are using the cell prep for downstream analysis for functional assays, then we might end up with fewer sorted stem and progenitor cells if they are over digested. So I think that's one critical step, being able to find the right time.
Yeah. So I guess you did a lot of experiments on that, right?
Yeah.
To
So
really pick the time, the correct time.
Right time, but also the cutting, of course, if the mechanical digester also needs to be optimal. Right? So we have optimized that.
So are the markers that identify myoblasts and muscle stem cell populations the same for mouse and human?
Yeah, that's a good question. I haven't yet been able to test the panel of cell surface marker on the human cells.
Mm-hmm.
So
yeah,
that's for the
it
future.
is challenging, right? Yeah, because you have probably you need like to create and validate a full new panel. So that's a lot of work. And I don't know, but is sample availability an issue for human
Yes,
studies? Yes,
I
right?
think that's the biggest issue. Being able to gain access to enough material for human muscle biopsies in order to test those panels. Yeah.
So think about that. Is there any organoid system that can be used for this, for muscle regeneration studies?
Yeah, I think yeah, they're being developed. The muscle organoids. There are IPS, of course. There are iPSC, induced pluripotent stem cells that could be used to test. But even then, it's about are we really looking at a sample to recapitulate the human muscle myogenic population, right? So ideally, we would like to start from needle biopsy from patients or from volunteers. So,
I see. So another question from chat. Does your work on understanding aging in muscle cells open up avenues for treatments, for example, after injury? I know you've mentioned the blocking THBS1. So how is this, like, is this close to the clinic? You think it requires, like years? What are your thoughts on this?
Yeah, so this work was performed in mice, and of course there is a long way and to take something from the from the research. We call it from bench to bedside, right? So it takes a really long time, and so further studies are needed to be able to test this model in human samples. And then from there, of course, we could then move forward and try to develop treatments. But yeah, it's it's far away from applications for now.
Is this something that you will, focus on your own lab, trying to move this towards the clinic, or you will focus on furthering the mechanistic understanding of all these processes? What are your interests here?
Yes, so the idea is really to understand how of course, this is an aspect that I'm very interested in. I think that it's really exciting that we can modulate a specific pathway, and then we can get improved regeneration in aged mice, which also leads to increased strength. And this has a great potential for therapeutic applications. So we have tested that we have shown that treatment with a blocking antibody leads to increased muscle strength in absence of injury, as well as in the context of injury. And so I think this is really exciting. And yes, I am looking to extend these studies to human samples. And
excellent.
Yeah,
good luck with this. Yeah.
Thank you. But and then another aspect of the lab is, of course, to study how immune cells are contributing to the regeneration process and how do they really interact with the stem cells to support the regeneration, and how is this interaction altered in the context of aging or also muscle diseases.
Yeah. Well, especially I guess with C47 being a don't eat me signal, I guess all the myeloid populations there have a lot of interactions on that regard. Do you have any thoughts on what is going on there?
Yeah. Well, of course, I didn't go into the details of that, but yeah, CD47 is a very interesting molecule because it's both a ligand and a receptor. So it is a ligand for SIRP alpha, as you mentioned, which is expressed on the surface of immune cells. And the interaction between CD47 and SIRP alpha leads to an inhibitory signal that blocks phagocytosis. And this signal is upregulated on cancer cells. to evade immune clearance. And it's also the receptor for thrombospondin 1, right, as I discussed. And so it is very interesting that the dysfunctional muscle stem cell in aged tissue, in the aged tissue, are actually expressing high level of CD47. It could be a way for these cells to really hide from the immune system and persist in the tissue and cause this dysfunction by producing the thrombospondin, which is an extracellular matrix protein, which will then act on the minority of the CD47 low stem cell, which are functional, then suppressing their proliferation. So I will be really interested in studying how the CD47I stem cells in aged skeletal muscle are interacting with SIRP alpha expressing immune cells and see whether can we actually empower the immune system to clear these dysfunctional stem cells. So I think that will be an exciting avenue that might synergize, for example, with thrombospondin blockade to enable improved regeneration. Yeah.
Yes, very, very exciting, especially if you can, show some of the interactions in the spatial context. That would be certainly amazing. Okay, I think we've covered, a lot of topics. So I think we can, call it off here. Renee, I think, yeah.
Yeah, thank you. Thank you so much for both an interesting talk, Melinda and Xavier, for great moderation. And thank you everyone for being here. Yeah, and I hope to see everyone again next time.
Thank you so much. Bye.
Thank you. Bye-bye.
Bye. Thank you.